Okay, let's start. Welcome everybody. This time not the usual faces, the usual hosts of the Hot Politics Lab, but me and Isabella are hosting this lab session today, mainly also because Gijs is presenting, of course. And yeah, most of you probably know Gijs, so I probably don't have to introduce him, but for who don't know him, he is an associate professor at the University of Amsterdam, the Department of Political Science, and co-director of the Hot Politics Lab. And his work now focuses mostly on political psychology topics such as personality and populism and rhetoric of political elites and using text analysis. And of course, a big topic he studies is emotion and emotional responses to politics. And he's going to talk about that today, and a super exciting project. I think it was data collection of maybe 4 or 5 years. So a lot of data. And I think Isabella and I both helped also collecting the data. So super curious to see what you did and what you found. And yeah, if anyone has a question during or after a presentation, there's a Q&A box below. So you can type your question in there, and then Isabella will read it out loud after the presentation. So Gijs, the floor is yours.
Thanks, Mike. And let me switch to my PowerPoint. And great to be presenting here for once. I'm going to talk about psychophysiology today, but particularly FEMG, so facial electromyography techniques, which aren't used much in political science at all. I think the number of publications, you can probably count them on one hand. And I think that that's a real pity, because it makes talking about emotions in politics very limited. Because most of the political science work on emotions is about survey questions, using survey questions to measure emotions, and that makes it essentially impossible to tap into any of the unconscious aspects of emotions. Also, something we know through comparing FEMG data to survey batteries on emotions, is that they don't measure the same thing. And this is not a matter of using the wrong techniques, that FEMG is not a valid technique, but rather that FEMG and survey questions about emotions measure fundamentally different properties. They all measure something that relates to the huge container concept of emotion. But any of you who've ever read a definition of emotion will acknowledge that it's a huge, a very wide concept that encompasses conscious and unconscious aspects of information processing, and it also covers a wide range of expressions and attitudes and postures, etc. So in retrospect, it was perhaps quite brave to assume that only with survey questions we would be able to capture this very rich theoretical concept of emotions. So I think that we should look at facial EMG data too as a complement to existing survey questions, because it allows us to tap into an additional set of this container concept of emotion, and thereby we can obviously learn more about the human condition. Okay, but why is FEMG not used a lot in political science? Well, first of all, it's quite technical, so that it clearly creates a barrier to those of you who want to use it. And I'm just going to say yes, that's true. There is definitely a barrier in terms of technique. At the same time, it is also a lot less challenging than, for example, engaging with EEG data, something we're currently doing. As you can see, Mike is actually in the lab waiting for a respondent to finish, I think, or is already finished. No, already finished. Okay. I've seen last week how they do that. Well, I mean, they have to actually connect 64 electrodes, and it compares to just 4 to 6 electrodes for FEMG. So that's already a huge difference. fMRI is probably even more complicated. So at that sense, your level of technical expertise is also a lot less demanding than with other work that engages with these unconscious aspects. Now, the second point is that it's difficult to process the data, and that's definitely very challenging. Luckily, there are a lot of recent developments in the field of psychophysiology that can help you create a better sort of workflow to process data. And what I try to do today is to contribute to that process too. And thirdly, a big challenge is how to interpret the data. If there's one important thing about emotions, it's that context matters, both in terms of what you show people, but also in terms of, respondent characteristics that may explain why some people see, focus on one thing and one thing in the treatment, and other respondents focus on another thing in the treatment. So we need to explore treatment characteristics and respondent characteristics in order to describe and understand what FEMG data can bring us. And today I will particularly focus on the latter. I will very briefly take you to what I call pre-processing hell, but then I will fast forward to what I would call benchmarking heaven. And in doing so, I'll present some sort of meta study. So this is indeed 5 years of data collection, 5 entirely different experimental designs with actually multiple designs in designs, many different locations where we collected data, a total of 585 respondents and 98 different experimental treatments. Now, clearly I didn't do all of that work alone, luckily. And so this is joint work with a whole bunch of people who through the years helped collect, process, and analyze the data. And so for those of you entirely unfamiliar with facial EMG, the picture here on the left illustrates this. So essentially what one does is placing electrodes on facial muscles. So for example, on the top of the head of this doll, there are 2 electrodes that pick up the activity of the corrugator facial muscle, which is activated in response to negatively valenced treatments, in theory. Then the zygomaticus muscle on the cheek is associated with positively valenced treatments. And then there's a third one that we have also done research on, the labii. This is the muscle next to your nose, which is associated with disgusting treatments. There are many more facial muscles that are also relevant in recognizing emotions, but we haven't particularly analyze these, so these are for sure the most common. There's also something called skin conductance that has nothing to do with FEMG, but with psychophysiology broadly. But I won't talk about that today. Okay, some of the work that I will be using in this meta study has been published. We currently have 3 publications on psychophysiology, and there's a bit more to come. But some of the things I will use today have not been published at all. So this is a brief overview of the 5 different designs I mentioned. So as you can see, there are 5 different designs, but we've also collected data at various locations. So particularly design 3, we've collected at 4 different locations. And in each of the designs, everything is different, essentially. So the number of respondents, the number of treatments here, that's the second N. The FEMG measures we took, so we always took corrugator, but sometimes we did zygomatic and sometimes we didn't. And these are the different treatments we had. So we have treatments where people see information on issues. We have treatments where people just see pictures, neutral pictures, or pictures of super gross and disgusting and weird stuff, like, I don't know, tumors growing out of the head of a baby, but also pictures of party leaders, pictures of people expressing a particular emotion, pictures of party leaders expressing a particular emotion. We have treatments with sounds, treatments without sounds, and so there's a whole bunch of treatment characteristics in this data set. Okay, very briefly, I'll take you to pre-processing hell. So when it was always, I mean, once we started to work with these wires, everyone was thinking, oh, that's really cool. And we were invited to lots of places to talk about it. But it was actually easy to collect the data. It took us much longer to properly analyze this. And this is a little bit because of pre-processing hell. And somewhere, I don't know, a year into the project, Bert Bakker started to say that he was reappreciating the Likert skill again, while having to work with this FEMG data. Now, we didn't entirely fall for this, obviously, but it does make you reappreciate sort of the very basic standard tools we as political scientists are used to work with, because they're so much easier to pre-process. There's barely any pre-processing involved, and for FEMG data, you need to do quite a lot of pre-processing, and I will particularly highlight here 5 analytical decisions that in the paper that is associated with this presentation. This is explained at length, and we do lots of simulations to show what matters and what doesn't matter. And the bottom line is that with processing FEMG data, there are a lot of researcher degrees of freedom, but luckily a lot of them also don't really matter. So that is helpful to know, I think, because when I started to read about psychophysiology and FEMG in particular, you can take 5 or 10 papers and each of them processes the data differently and it calculates the DV differently. And sometimes that's because of fundamental differences in the treatments, like a very long treatment or a very short treatment, but sometimes it also seems to be quite random. Same authors doing something different every paper. And so we are trying to bring a bit more system into these analysis. And so, okay, let me just briefly walk you through this. So the first thing, and that is common to everyone, all of the FEMG papers, is compare activity in the treatment to individual baselines, because activity between respondents can differ dramatically for reasons that have nothing to do with your experiment or nothing to do with the respondent. It could, for example, have to do with the quality of the wire you're currently using. So you don't want to have things like that to play into your analysis. So that's why you're comparing individuals to baselines, to their baseline. So there should always be somewhere in the experiment, there should be a period where you measure this individual baseline, and ideally you do this every time before a treatment. Now, how to calculate that individual baseline? There's a lot of variation there, how people do that. We had student assistants code basic tendencies and baseline activity, and then try to correlate that to all kinds of different calculations of the baseline. And well, for a very long story short, it's actually quite efficient just to take the median of activity in the baseline as your reference point. That's a very simple solution that that you don't have to argue much about, and it correlates very highly with what these coders identified as the general tendency. Then we divide treatment activity by the baseline activity, so, and then times 100, so that your scores are always a sort of a percentual increase in active FEMG activity. Now, there are a lot of different ways to do this. In the past we used subtraction from the baseline. Some people use Z standardization, and well, in the simulations we did for this paper, we find that these choices don't really matter. Now, thirdly, what is important is that we use multi-level analysis to analyze treatment activity. And this is a big difference compared to what people were doing, I don't know, up to 3 years ago. So we're not the only ones who suggest this. A recent paper by Ursula Hess who also suggests this. The problem with FEMG data is that there's always an initial response to a treatment. Well, if there is a response, then there's a peak in the beginning that then regresses back to the mean or regresses back to the baseline. But FEMG data also declines over time. So there's sort of a slight negative effect of FEMG activity throughout the whole experiment. So it might actually be that within your treatment, there's an initial response, but then the regression to the baseline is so severe that the mean of the treatment is actually 0. And so we run a series of simulations assuming different effect sizes. And at one point, especially with the smaller your effect size, you actually risk rejecting the null hypothesis if you take just the mean activity over the treatment. So that's why we propose to not analyze mean activity, but to analyze second by second activity and use a multi-level solution where you add the respondent as a particular level. And then you can also model specifically the time dynamics of that respondent within the treatment, which is something you also can't do if you just look at means. Now, regarding pre-processing, there are lots of different things you could do. What we did was we had coders look at individual plots and tell us whether something looked really weird. So for example, there could be cliffs in the data where there's suddenly a big drop or a big increase in activity. There are people who just don't seem to respond at all. Their values are very low, and if they change, it's just between two values. So we coded that, and yeah, we also coded any additional things that look weird, and this went pretty well. So the Krippendorf's alpha, so the agreement between these coders was pretty high. But also in terms of effect sizes, we measure, at least for the corrugator, we quite quickly reach a level where the coefficients don't change much anymore. So what I did here is I'm comparing FEMG responses to a tumor picture, a picture of a tumor, to a picture of a very cute baby. So the effect size should be positive for corrugator because, yeah, tumor looks, ugh, and then the baby looks, oh, that's sweet. So what you see here in the first coefficient is where we did no pre-processing at all. We just put in all the data. And then we progressively took more and more things out. So first we take all the cliffs out, then we take the flats out, so the non-responders. The two categories after that are basically whether there was agreement between both coders or just one coder identifying it. And then the last two, we furthermore, after removing these visually inspected outliers, we winsorized the data, and in the last category, we also throw statistical outliers, so more than a deviation of more than 4 standard deviations from the mean. But you see with corrugator that the effect is pretty much the same. It's a little lower for the winsorizing. And obviously, with each progression of pre-processing steps, the standard error gets smaller. For the zygomaticus muscle, it's quite different. It's a very difficult muscle to capture. There's so-called crosstalk. So if you sort of pull the muscles on your what's it called? Your here. Jaw, that was the word, the muscles on your jaw. It might be that influences the activity measured in the electrode on the zygomaticus muscle. So there you see a lot more variation, and only when we get to winsorizing or throwing the outliers out, we're able to capture the statistically significant effect of lower zygomaticus activity, because you should smile when you see the baby, right? Not when you see the tumor. Okay, now let's get to benchmarking heaven. So the 98 treatments we have, we can code them following a range of different treatment characteristics. So is it about politics or not? Is it a video or an image? Does it show if, this is only for the political treatments. Does it show a face of a politician, or does it concern the description of an issue position? Is it positive or negative emotions that are part of the treatment? Is there audio or not? And was it measured in a lab or in the field lab? And then we have a whole bunch of respondent characteristics that I've just listed here. And so what I'm going to do in the next few slides is to take all this data we have and then throw in one treatment characteristic and describe whether it has an effect or not. And after having done that, we're going to do the same, but then also throw in the respondent characteristics so that we get to know the context more. So these are treatment characteristics. So these are just multi-level models with the whole specification I talked you through. So we took the median baseline, we divided activity by the baseline, and then I calculated two different effects. So the outlier removed and the Winsorized. So the last two options in the pre-processing figure I showed you before. And these are all individual analysis. So I didn't, I threw in, say, one variable to measure political versus non-political and nothing else. So it could be that some of the effects here are actually explained by some of the other treatment characteristics. That's something I still need to figure out. Okay, so let's look at this. Let's look at the top row, the corrugator analysis. What I think is some of the effects look really small, and they're probably small, but yeah, it's also the pictures are a bit blown up because of the quite high standard error on some of these coefficients. So these are regression coefficients with their 95% confidence intervals. So there is a very small but statistically significant effect of political treatments. So political treatments are actually receive more corrugator activity than non-political ones. And remember, some of the non-political ones are really gross and should really have been selected because they actually evoke a strong response. And we replicate that. I mean, it's from other studies. So the fact that these political treatments receive an even higher response is, I think, quite telling of the fact that, politics is hot. So we can still keep our lab's name. I mean, for a while, we were worried that we would have to call it the cool politics lab because we weren't finding much. That was, I think, because we were still in pre-processing hell. All right, what else do we see here? What is very nice is that there's no difference between the lab and the field lab in the corrugator. What you see also is that negative stimuli provoke more corrugator activity than neutral stimuli. That's what you expect. Stimuli with video or with sound are slightly more stimulating than videos without. And finally, and interestingly, the phase effect is negative. So issues actually provoke a bit more corrugator activity than phase. Now for psychomaticus, things are a bit more complex. There is definitely a difference between lab and field lab, for example. And some of the effects actually skip from below the zero line to above the zero line. It's probably because there's much more variation there within these dummy variables. Each dummy variable summarizes a whole range of different treatments, right? So this is still something to figure out. Okay. Now, regarding the respondent characteristics. So what I do now is I only take out the political versus non-political variable, and I interact it with different respondent characteristics. And I don't have a lot of time, so I'm probably going to skip a few and focus on the most interesting ones. So let's start with political knowledge because that's quite interesting. So you see here on the left the corrugator activity across political knowledge. So this is the interaction effect. And so on the left you see people with low political knowledge, and then and on the right people with high political knowledge. The red line is the regression line for corrugator activity in response to non-political treatments, and the blue line is corrugator activity in response to political treatments. And if you look to the left, you see the same, but then for psychomaticus activity. So what and you see a few differences here. The confidence intervals are quite wide, but these differences are statistically significant. And what we see is that people with low political knowledge have more corrugator activity in response to the political treatments than the non-political treatments. And people with high political knowledge have more psychomaticus activity in the political treatments than the non-political treatments. And this difference is exactly the opposite for people with low political knowledge. So Yeah, that's something interesting. I want to reflect a bit more on this in the last slides. Okay, alcohol consumption, I'm going to skip. This one I'm going to skip. Cynicism doesn't do anything. That's interesting. Political interest, there's also a clear difference. So the more politically interested the respondent, so if you move to the right on the y-axis, the stronger the corrugated response to the non-political treatments. But if you look at psychomaticus activity, it's reverse. So the more politically interested the respondent, the stronger the psychomaticus response to the political treatment, so the blue line. And again, I will try to rationalize that at the end. Vote choice, I made 4 groups because we have so many parties that just didn't fit the screen. So I summarized them in left-wing, center, right, center parties and right-wing parties. And then other are people who said that they don't want to say, or they don't vote, or they don't whatever. And here, most groups are quite alike, except for the right-wing respondents. So they respond more strongly to the political treatments, while having weaker responses to the non-political treatments. And by the way, I skipped the left-right ideology moderator. These findings aren't exactly the same. That's what I will say. So that's a bit of a puzzle I still need to figure out. Yeah. For psychomaticus activity, there is not much of a difference between voters. Oh, wrong button. If we look at education level, you see that political treatments evoke more corrugator activity, particularly among people with secondary vocational education. So that's the one here there. And the rest is more or less the same, but that's quite interesting. So these are people for the Dutch people among us who've finished. So it's about finished education level here, who finished MBO. And something like university graduates, so people have actually finished university, there's really not a difference between the 2 treatments. Now, if you look at psychomaticus activity, we again see the biggest differences are, well, not there, sorry. So the university graduates, they're pretty much the same, but it's mostly the people who finish secondary vocational education or higher vocational HBO education. And so they are, again, they are more responsive to the political treatments than the non-political treatments. Okay, so I guess this went all pretty quickly. What do we actually take away from this? Well, I think one of the first things that I think is really important to see here is when we need to create benchmarks. So, for example, in the study I did with Maaike on disgust, we benchmarked the disgust the disgust response that people had towards seeing an out-party leader to a picture just showed someone vomiting, right? So and what we found was that people are equally disgusted by seeing the out-party leader as by seeing someone vomiting, right? So I think that's a nice way of sort of setting the nice and disgusting way to aligning political treatments in sort of the larger universe of treatments. And so what I think I was really happy to see that on average there's a very tiny positive effect of political treatments on corrugator activity. So so political treatments really do impact people. But also what we saw in those last few slides is that there are lots of different respondent characteristics that explain differences in responses to political versus non-political treatments. So the context, the context of the respondent actually seems to matter. And there seem to be in particular 2 routes. So one is that people with low knowledge or secondary vocational education, low knowledge, I mean political knowledge, and right-wing voting is associated with higher corrugator activity in the political treatments. The other route is high knowledge, high interest, and higher vocational education is associated with higher psychomaticus activity in political treatments. And psychomaticus for us has been a bit of a mysterious response, because in many cases our treatments aren't funny at all, but we do see a lot of psychomaticus activity, especially in places where we didn't expect them. So one of the studies that Mike has also presented in the lab, she reported that there's quite a lot of psychomaticus activity in response to an out party leader. And so the psychomaticus muscle seems, well, that's our interpretation, seems to be involved much more in things like contempt than in terms of a real positive emotion. So what the second root here that I describe may hint at is sort of a root of contempt. So it's sort of the higher educated who express this, whose faces express this contempt in response to particular political treatments. Of course, we can investigate this more by subsetting towards the cases where they should show contempt. And I think, this is the last slide. So I want to thank all of the people involved in this project, in particular the Big Eight, and I'm looking forward to questions.
Okay, thank you, Gijs. Just as a reminder to everybody, if you have a question, please type it in the Q&A box and I'll read it out loud. And we'll give you a few minutes to type your questions. Let's see if we have. No, maybe I'll start with a question, Gijs. I was wondering, you opened your talk by talking about context in politics and how it matters. And then one of the distinctions that you make between your treatments is between negative and positive. And I'm wondering how, especially for political treatments, isn't whether something is positive or negative up to the respondent to decide. I can imagine with the political treatments, it's very clear the baby is positive, the tumor is negative. But for political issues, isn't it up to the appraisal of individuals? And did you account for that or analyze that somehow?
Yeah, thanks. Do you hear the van on the background or? No, okay, good. My daughter is giving me some fresh air. No, we did not account for that, particularly not in the analysis I've just shown you. So it's just looking at the difference between political and non-political treatments. So indeed, there is still a lot of heterogeneity in these effects. That all the more surprising, I think, it is that we already find some of these results. I think on average, I think I kind of understand why we find this second root of the psychomaticus activity with the higher educated, because on the out party effects that Maaike finds are much stronger than the in party effects. And also what Bert, Matthijs, and I did in the Hot Politics paper is that the incongruence effects are much stronger than the congruence effects. So this is probably why I find these findings, is because we haven't split it out yet to whether this is indeed the context of a positive or a negative context for the respondents.
Yeah. Okay, we have some questions now. The first one from Diamantis. He says, thanks so much, Gijs. This is a very interesting topic. I'm surprised by the sound-no sound comparison that looks rather insignificant and small. Music is known for eliciting emotions, but I guess that facial EMG wouldn't tackle the presence of these emotions. Did you also look on the psycho-physiology data, like EDA? And do you have any other idea about why you might have not found differences?
Let me just pull up the. Okay, yeah, there it is. So there is a sound effect, a very small positive effect. The reason why it is so small could have all kinds of reasons. So maybe if I, for example, add another treatment characteristic there, this sound effect may be much larger. So that's one reason. Another reason is that there's actually a bit of variation in the treatments that have been coded as having sounds. So this is a very general label. So there are the issues who have music, voiceover, everything. But then there also, we, for example, also have the moral violation treatment, where there's also a voiceover that says, this is Mark Rutte and he took bribes, blah, blah, blah. And then there's another example where we had words, just words in the screen with a voiceover. So one of the words would be bill or healthcare. And so, yeah, I think there's probably quite a lot of heterogeneity in that sound variable, and we can't explore everything because it's not sound is not randomly associated with treatments, right? So it's particular treatments that have particular types of sound. And you also asked about EDA, electrodermal activity. We have all the data there, so I can do these analysis for SCL2. No problem. But I think, and I think we discussed that in the past too, but if not, then I think what would be more helpful if we would really be interested in analyzing sound is to take the issues, because the issue treatments, and then analyze the second the sound sets in, because it sets in prior to the language, the rhetoric, and prior to the videos. So, yeah, very long answer, sorry.
Next question is from Stuart Soroka. Your overall results for the corrugator and the sigmaticus are in line with past and now slightly discounted results, suggesting more negative responses for conservatives. Is that a correct interpretation? And how do you feel about that?
Yeah, so actually then I want to show you also the left-right ideology question. Yeah. So this is a bit weird because they're pointing in different directions and I haven't had the time to figure out why. So yeah, thanks, Stuart. This is of course a great question. So what if I take left-right ideology, which was just a very simple question, just place yourself on the left-right ideology scale. So what we find here is that left-right ideology matters for non-political treatments, but not for political treatments. In the sense that the right here has a slightly higher corrugator activity to the non-political treatment than the left. But the blue line, those are the political treatments, is basically a flat line. Psychomaticus activity, then yeah, actually shows the same the same pattern. And then if we switch to vote choice, hips here we see that the right actually has more corrugator activity. So this is based on vote choice. The label is wrong here. Sorry, it says political interest, but it should be vote choice. Here there is actually much stronger response of right-wing people to political treatments. So this is and a weaker response to non-political treatment. So this is puzzling. This doesn't make a lot of sense. So I kind of need to look more into this. But I think regarding the work that you refer to, I think I've always felt about one thing. I felt particularly ambivalent that that conservatives have been hypothesized to have stronger responses to threatening information. And that was strictly operationalized by looking at arousal. And so this is not arousal. So in that sense, this is a different finding. But always in reading that material, I've always been thinking, are they really talking about arousal or valence? It wasn't so clear to me. So you might have well have hypothesized the same thing and then look at valence. And then then the ideology question does suggest that to non-political treatments, the right is more responsive. But then this suggests something else. So I don't know yet. There's probably quite a lot of variation at the level below the simple dummies that describe these treatment characteristics. But actually in our paper, the Nature Human Behavior paper, we also analyzed somewhere in Appendix C 47 or so, we analyzed the set of pictures from the original team, that's also in here, and took the corrugator as a dependent variable, and there we didn't find a difference.
Okay, the next question is from Philip Mendoza. Super cool presentation, Gijs, and also very interesting to see these aggregate thoughts. Considering the threat effect for people who don't know as much about politics or are vocationally educated, I was wondering whether this could be a result of mistrust in elites or anti-elitism. Have you already considered this and included such items in the study so far?
Yes, we did. That's cynicism. I should say political cynicism, really. So this is based on 3 questions. What are they exactly? Politicians can be trusted. Politicians are profiteers. And then a third question that is very similar. And we took, we just added that together. But yeah, as you can see here, these lines totally overlap. So there's not, at least, maybe they do something else, but it's not that cynical people are more responsive to political treatments than non-cynical people. That's not what we find. And we also actually have a very particular populism treatment in our data. And so far, yeah, we also don't really find an effect of cynicism there. So yeah, that so far doesn't seem to be the story exactly.
Next question is from Tobias Rohrbach. Super cool overview and results. You emphasized the relatively small effect sizes several times. How does the size of your effects compare to the usual effect sizes from physiological data found in the literature?
Well, I think one of the problems that I have with psychophysiology data is that they've done so many different things, particularly in how to construct a dependent variable, that I would find it difficult to say what is an average effect. But I can, of course, contrast these effects to to sort of the standard deviation in our data set, right? So, and also, let me make one footnote here. One of the problems with trying to apply lessons from psychophysiology to political science is that a lot of the psychophysiology work is interested in very specific subsamples. So people with specific pathologies. And so I don't think that work should be used to create a benchmark in terms of what sort of effects are considered to be small or large. Now, what I've said in there's a pre-registration plan for this paper where we only pre-registered the sampling and analysis plan, so there are no hypotheses. And I pre-registered that an effect B is 20 would be a large effect. Why? That was the effect size I found for corrugator activity in terms of the difference between the tumor picture and the baby picture. Right, and those have been selected because people have found corrugator effects there, and so we find an effect of a 20% increase in corrugator activity compared to individual baseline. In terms of the total standard deviation in the data set, 20 is something like 2/3 of a standard deviation. So it's quite high. Some of the effects that we find here are, let me go up. The respondent characteristics, you see actually only this negative thing approaching 20. So that's all the others are really tiny effects of just 1 or 2 points. Yeah, those are, in terms of standard deviations, yeah, tiny effects. 1/10, 1/15 of a standard deviation. But these moderator variables, yeah, sometimes they travel about 10 points over the entire range. So that's half of the tumor effect. And let's see the vote choice. Yeah, here. This actually isn't too large an effect. It's just something like 5 or 10 points again. But here I thought, oh no, this is about the same. Okay. The Psychomaticus effects are a bit larger, but also the standard deviation there is larger. So I haven't done the exact math there, but I think it's comparable in turn if you would express it as a standard deviation of the total, as a fraction of the standard deviation of the total increase.
Can I ask you a question, Isabella?
Sure.
Gijs, can you, based on your experience with these measures and going through the pre-processing you described, what are your recommendations or your key takeaways for someone who would be interested in doing that, in terms of learning to take those measurements, recommendations on how to store the data? I think this is quite large in size in terms of data collection. How to deal with the data? What are your best practices for someone who wants to learn that?
Yeah, you're totally right. I mean, this data set is a couple of gigabytes, but I think here it might also be the particular software you use. So we've used a program called VSRP98, and 98 stands for the year in which it was developed. So you can kind of get a sense for how incredibly modern, up-to-date this software is. And so I'm pretty sure that the BioPack software, for example, has a more efficient way of storing this. Yeah, but the point why it's so large is that I've analyzed second by second, but in principle, it depends on your what's that called again? Well, you can essentially set the level at which you want to collect data, and so typically you would actually set a much lower level, something like 100 or 1,000 of a second. And so it will collect all of that. So you can imagine that having an experiment for an hour, that for each individual is already a substantially sized data set. So yeah, what would I recommend? Yeah, I think I mean, the one thing I would recommend is that you have enough time. It's incredibly time consuming to do this, but I think it's important. I mean, one of the benefits of doing it now in compared to 5 years ago is that people are getting more serious about the reliability and reproducibility of FEMG results. So different research groups are working, writing papers or doing pre-analysis papers where they describe in much more detail what they are doing and why than you were reading in sort of the textbooks that were out there 5-6 years ago. I mean, there's literally a textbook who says, well, baseline period, yeah, you can take something like 5 or 6 or 7 seconds off the baseline. Why? I mean, it seemed to be totally random and a little bit results driven, maybe. I don't know. And so what I can advise is a few things. Yeah, so use coders to go over all of your data to see whether they pick out anything weird. Write a pre-analysis paper, but don't think there is just one proper way of going about it. There isn't. I mean, how are you going to tell the difference between a huge response and an outlier? You don't know. So what I'm recommending, so what I did here was like, I take two measures that follow a slightly different procedure and strive for robustness. And so you also saw, for example, that some of the findings weren't robust. For example, so positive neutral here actually has a positive effect here and a negative effect here. So this effect isn't robust. And so on the basis of that, I would accept the null hypothesis. Yeah, and what more? Yeah, I think I'll just leave it here. It's just could probably talk the whole day about this, but yeah. But I guess the last point is the key one. Is to strive for robustness and don't pretend there's one unique way of doing it.
Maike, I think you also had a question.
Yes. Yeah. Thanks, Gijs, for this really cool presentation. I was just wondering about the length of these treatments. So you found no difference between videos and images. I think the videos were mostly longer. Yep. And I was just wondering, because you also use this multi-level modeling, but do you in general see some sort of like within a couple milliseconds or seconds sort of a peak, or is it during one treatment some sort of just general higher activation? So I was just wondering, how does this response look like? And also maybe how long does your treatment need to be in order to really pick up a response?
Yeah, so I've looked, I mean, for a lot of treatments, we have this 8 seconds exposure time. And I looked a lot at different responses to pictures with these 8 seconds. You don't find that people only respond in the first 2 seconds. Yeah, some people do, some people don't. And you know, we can't, yeah, I mean, there might be all kinds of reasons. Maybe they were momentarily distracted or something. Maybe it took a bit longer to process what they were seeing. So in that sense, I also particularly don't recommend to take out parts of your exposure time and only use that as your analysis material. Then comparing treatments to images to issues or videos. The videos are indeed much longer, on average something like 45 seconds. And there you do see, yeah, you see a bit of, depends on the respondent really. On average, there is sort of a long-term decline, but it also happens that in the middle of a treatment, there's suddenly a response that really depends on what you're showing them. What is changing. Now we've also, for one of the data, actually the exposure times is varied, but I haven't yet analyzed that. So we actually have these group of leaders that were shown either 20 seconds or 2 seconds. But so vaguely in my memory, I remember that didn't matter much, but yeah, I think that's all I can say currently.
Maybe I'll ask one last question, and that's a bit of an overarching question as well. And so I think the hope was to pick up with the FEMG measurement also valence and not only arousal, which skin conductance can do as well. But then you argued that actually the zygomaticus is also activated when people feel contempt, which is also conceptually related, I would say, to disgust. So then maybe this is a mean question, but my question is whether FEMG picks up something other than arousal, and why should we do it if maybe we could also just do or measure skin conductance response, which is, I think, a little bit easier.
It's definitely easier, but this is not arousal, right? This is valence, and I think, look, ideally, with FEMG, you would be able to pick up discrete emotions, right? So contempt, you know, following this action unit coding system, is both the psychomaticus activity and this Dimpler, I think it's called, right, Maike? the muscle here below your lip. So in that sense, we shouldn't be too surprised actually that psychomatics activity seems to be associated with things that can be considered contempt. The problem is really that, but nobody really uses it, FEMG to that extent, I think. It rather boils down to positive versus negative. But that I think is strictly because it has only been validated to measure positive versus negative. And so I think there's actually more to gain from it, but no one has shown that works yet. Yeah. And regarding arousal, and in the end, I sort of want to include arousal here as well, but just on the basis of the results we've had in these other studies, where SCL wasn't typically the main goal, but they were always quite different than the valence results. And it's not as if there was a correlation higher than 0.1. And so I do think they measure something structurally different.
Okay, thanks, Gijs. We are at the end. So I think this was next week we have one more meeting and then we're actually done for this semester. So next week at 2:30, so a little bit earlier than usual, a half hour earlier, we have 4 of our interns from Brain and Cognition and Neuroscience who actually collected some data, EEG data. So they hopefully will show some results and definitely talk about their projects. So really exciting. So definitely tune in for that. And for the rest, we will be back, I think in September, and we'll send around our program then, of course, as well. And for now, thank you, Gijs. And I cannot really give you a hot politics mark because you're the one who owns them or has them. So
I have enough.
You have enough, I think. So thank you very much.
You're welcome. Thanks for all the questions.